There are other factors which affect the plant productivity in the field, that is biotic factors. Biotic factors include the invasion from insect pests, the pathogens like fungus, viruses, and bacteria. All these pathogens, they cause diseases and result into a loss in the productivity. Similarly, insect pests, they are major factors which influences the crop productivity in the field. A lot of efforts have gone in to control the invasion from insect pests, and I'm going to focus on how this problem of loss in productivity by insect pests has been dealt with so far. So traditionally, the initial efforts were to find out insecticides, chemicals, insecticides, which successfully were discovered and were used in the field. The problem with chemical insecticides and the pesticides are that they cannot be degraded in the field. Once they are spread, they remain in the soil, and then they find their place into the chain and reaches to humans, which are toxic. So this factor had been there. And also, these chemical insecticides are broad spectrum. Once they are spread, they not only influences the specific insects which harm the crops, but also the insects which are not harmful to the crops, but they are also killed by these insecticides. So there had been an effort to develop insecticides which are more specific for a specific insect type which infects the crop plants, or specific crop plants. So in this journey, the use of a bacteria called Bacillus thuringiensis was discovered, and we are going to discuss this aspect in greater details today, including its history, and then the usage of this bacteria in good old days, and then how the studies with this bacteria led to the discoveries of an specific insecticidal protein which this bacteria produces and is very specifically targets a specific insect type. So all these aspects, and then how the concept was built that instead of using the whole bacteria, why not we make plants to make that protein which is toxic to insects, and when the particular insect eats that or feeds on the crop plant, it is killed without further damaging the plants. So we are going to see this this aspect in today's lecture. So, insect resistance using BT technology conferred to the plants. So, let us look at what is this bacteria to begin with. So, Bacillus thuringiensis is known for over 100 years now, and it's a gram-positive aerobic bacteria, rod-like. You can see this; they look like this, and motile, and they sporulate, and They occur, they are soil-borne bacteria, and they are found everywhere, in the world. And they produce a, crystals, or what we call endotoxin, or these endotoxins are called cryproteins or delta toxins, which are toxic to insects in their larval stage. In the very early stage of insect development, they are harmful. Okay, so this bacteria is known for over 100 years. You know, uh, so what happens when this crystal protein um, is ingested by bacteria and reaches to stomach? It's basically stomach poison, and once it is eaten by the insect larvae, they stop feeding, and in due course of time, they die within 2 to 3 days after feeding on to this protein. So basically, it is a biological insect control agent, or in literature, they started referring to it as insecticidal crystal protein, or ICP. So now, when it comes to specifically rice, how it harms how the insects harm the rice plants, for example. So this is, if you look at the life cycle of the rice leaf folder insect, or the rice yellow stem borer. These are the two insect types which affects the rice plants in field. So they lay eggs into these leaves, and then the larvae, and the larvae eat the walls of the growing plants, and they come out as a adult fly, like this, destroying the plant. So if you look at the field, this is what happens. You see these white-headed plants, which are infected by the insects, And similarly, if you look at this stem borer, how it does, this is the entry of the larvae into the stems of the plants, and eating the stem tissues and destroying the plant. And you can see these infected plants in the field. And once they enter into the field, they destroy the crop completely. So that's how these type of insects, they affect the crop plants, very specifically the rice plants, on which I'm going to focus in a short while. So how this works? I mean, the Bt toxin, which is produced by the bacteria. So first is this Bt toxin is to be ingested, and then solubilization and proteolytic activation, and then it binds to the target site, and then formation of toxic lesions. So this is how, based on a lot of research is done, the mode of action of this Bt protein or crystal protein or the endotoxin protein was studied, and we will very briefly look into this. So basically what happens, when this is the endotoxin cryprotein, when it is ingested or eaten by larvae, it goes to the midgut area where a lot of proteases work on it, and it is chopped off into smaller protein molecules. And it is these smaller crystal proteins which are toxic to the uh, insects larvae. So from inactive endotoxin to active endotoxins are produced uh, by the action of proteases in the insect larvae midgut, and then these active proteins, they act on the target cells of the midgut area, and they kill those cells and create a hole basically in the midgut membrane, you know, and and then the larvae stop feeding, and eventually it dies in 2 to 3 days because of the damage is done to the midgut part of the larva uh, and of the developing larvae. So if we look at more closely the whole event. So if this is the crystal protein, BT crystal protein, it is ingested by the larvae. So it enters as the complete protein, which then solubilized here and then acted upon by proteases, and then the active toxin, acts here in the midgut area, like here the cells, insect midgut cells, they attached here to the cell membrane through specific uh, receptors, and they enter here and destroy these cells. And by activating the cell death pathway, and ultimately leads to the cell death. And pores are created. Pores are basically it stops larvae to eat, and then the larvae dies with prematurely without, I mean, getting developed into whole fly. So that's how this BT technology works. So the key point here is, if we look at the historical part of this, uh, BT was used as a pest control agent, that is the whole bacteria, the solutions, the bacterial solutions were spread, and they get colonized onto the plants, and when the larvae eats, they are killed. So from whole use of whole bacteria, then scientists try to find out what is the toxic part. So those studies revealed that it is very specifically a protein which they told crystal proteins, which are toxic to the developing larvae. And then the genes which were coding for this protein were isolated and characterized. And now we have a series of BT, ICP, or insecticidal crystal protein genes, which very specifically targets a specific insect types. So then the idea is that if we make plants to produce these proteins in the plants itself, rather than spraying the solution from outside. So that's where the use of genetic engineering came into play. That is, if you have the gene in hand, you can transfer this to the plant under appropriate promoters and terminators. And when this protein is made by the plant and the larvae eats that tissue of plant where it is being made, then it dies. So this was conceptualized after the discovery of these genes, which are making crystal proteins or cryproteins, what we call, and after it was possible to transform plant cells and generate transgenic plants, then this whole thing was merged together. That is um, the genetic engineering of plants expressing ET genes in enough concentrations, because that is very important in this, that the plant cell produces a certain level of toxic protein, otherwise it doesn't work. So whenever one is using this technology, we have to use appropriate promoters so that, so that it is toxic. The whatever protein is made, crystal protein is made within the plants is toxic. So all these factors were taken into account while constructing the expression cassettes. So the appropriate promoters were used to give high level of expression to make enough proteins. That's one part, and second part was the native gene of uh, BD, or Bacillus thuringiensis cry genes from because the codon biasness. Now the bacterial genes could not be expressed in the native form, and therefore that was modified. So what is in use today is the codon modified genes, which express in the plant cells using plant cell machineries. So promoter and the codon modified gene are the key to this technology, BD cotton technology. Okay, now coming to the BT rice. What is this BT rice we are talking about, or BT cotton for that matter? What it means is the rice plants which are expressing the BT cry toxin in them to fight against the pests, insect pests like caterpillars, moths, and butterflies. And since it is a natural pesticide, it was decided that the gene can be inserted into the plants, into the plant genome, and it is fairly safe. Okay, so when we talk of BT rice, we are referring to rice plants expressing cry genes in them and making cry protein, which is now. What are the benefits if we look at the benefits of BT rice? Now, this is the advantage is the use of insecticides in the field. That load is decreased here. You know, the use of pesticide is decreased. The amount is needed to be used in the field is reduced. You know, and the cost is not much between the BT and non-BT things, and it doesn't harm living creatures other than the targeted insects, and it is eco-friendly. So there are a lot of advantages using this technology, to improve rice plants, to improve their productivity. As I mentioned at the beginning, that insect pests are the major factors which damages the productivity in the field, and rice being the staple food for more than 60-70% of the global population. So it is very important to improve this crop plan against insect pests. So again, an example I will give the work done in my lab. Basically, we try to improve rice for this trait, that is using BT technology, particularly cryogenicid gene, to give resistance against a stem borer insect. Very specifically, the cryogenicid works against these stem borer insects. So The methods which are done is the particle bombardment system, and what it requires is basically a tissue culture protocol, which gives from tissue to the callus, and then from callus to the whole plant. You know, this needs to be in your hand, and then you should have the Bt toxin gene from bacteria coated with the particles, gold particles, and these gold particles are used through this machine, and they are bombarded onto the to the callus here, and then these bombarded calli are selected on selectable marker media, that is antibiotic, in presence of antibiotics, and then this selection pressure, in fact, continues until the plant reaches to the pot level. You know, until otherwise it will there will be leaky plants where where non-transgenic plants will also grow. So selection pressure of antibiotics need to be there, until the plant goes to the pots. So this is the sort of a sort of outline to generate transgenic rice, or for that matter, any plant. You should have these protocols in hand, how to transform. A lot of things need to be optimized in this machine. It's not that simple. And this also need to be optimized, the regeneration protocol for any plant on which one is working. So now you may ask why particle bombardment system. That is, the work in my lab was done well before the agrobacterium was demonstrated to be working with rice. So rice was in early 90s or in 90s was transformed successfully using physical methods of gene delivery like the particle bombardment system. And we developed this system and used it effectively in our lab to generate transgenics in rice for more than one purposes. One purpose I discussed in my previous lecture, and then this is the second usage. You know, so uh, so this is the plasmid constructs which were used. Here you can see ubiquitin promoter. This is a constitutive promoter, and you have the cryogenicity gene coding region and the NOS terminator. This is the plasmid containing gene of interest, and then you have another plasmid which has the selectable marker gene, in this case hygromycin phosphotransferase gene. It has a 35S promoter, and then you have NOS terminator, and this is the intron part of shrinken gene from maize. Now this intron is edited between promoter and the coding region to have more expression, basically. It was shown that if the coding region and the promoter is flanked with this intron, or this intron is flanked with promoter and the coding region, then the expression levels are higher. So these two plasmids were used in this work which we did, and this is the transformation protocols which were followed. Those plasmids were coated onto the gold particles and then bombarded, and the selection was made on hydromycin containing media. And like previously you saw that when the bombarded calli grown on selectable marker, you see this kind of scenario where white and black regions, these are the living growing cells, and all the black are the dead cells because of the antibiotics. And from here, these white patches were transferred to the shooting media, and shoots were regenerated, then rooting media, and then the whole plant seed set was open. So this is the optimized protocol for a regeneration of calli to the whole plant in the pots and the greenhouse, basically. So once you have these transgenic plants, they need to be, confirmed for the presence of transgene, and that is concerned, that is confirmed using southern analysis. So this is the southern analysis of of the plants. So you have control plant, non-transformed, and you have the transformed plants. Genomic DNA was isolated, cut it with the appropriate restriction enzymes, run it on a gel, transplanted on membrane, and then probed with the coding region of the gene. This is the positive control. This is the genomic DNA of non-transformed plant, and this is the DNA of the transformed plants. And you can see multiple copies getting inserted into the plant genome. This plant line has 3 genes, and this is 2 genes basically. And you can see clear-cut presence of transgene in the genome. And then it was. It needs to be confirmed whether the transgene entered is expressing, making RNA, mRNA, and also the protein. So the expression of gene was checked using another analysis. You can see the RNA from control plants. Control means non-transformed. And then you have the RNA from the transgenic lines, 2 transgenic lines. And the methods followed is the same. RNA was run on gel, transplanted, and then probed with the coding region of the trivalent gene. And you can see clear-cut hybridization of probed RNA to the genomic or the cellular RNA. So indicating that the genes entered into the genome are expressing, making transcripts, and the further translation of these transcripts into protein was checked through Western analysis. And you can see these are the 2 lines, and this is the control where the cry-specific protein were detected using cryogenesis-specific antibodies through Western analysis. So it means the plant, the lines which were generated were having the transgene, cryogenesis transgene. It is expressing, making transcripts, and also it is making protein. Now, as I mentioned in this, the bottom line is how much protein it is making in the plant cells, because you need to have 0.25 or 0.2 and above concentration of the protein per milligram fresh weight, which is required to kill the insects. So we did an ELISA to find out how much protein is made here. So these are the plants from the control plants, non-transgenic plants. So there is nothing, no protein here. And these are the 2 lines, which had 0.15, 0.19 in stem and leaves, at different stages of development. Booting stage is very, very early, and the tillering stage is the mature stage, where you can see the concentration is pretty high, 0.023 and 0.03 percent per milligram fresh weight, at this stage. And similarly, in this line, you can see it is pretty high at this stage, Tilling stage. Tilling stage is the one when the inflorescence starts for seed production. So it means the lines which we got are making enough of the crystal protein. Then the question was to be tested that whether it is killing or it is sufficient to kill them in the insect pest. So these insect bioassays were done, and that's how it happens. When you leave the larvae onto these stems, they enter, they create the black hole like this. So this is the entry. This shows the entry of the larvae into the stem actually. And once they feed on it, the whole thing becomes black actually. It is the dead larvae here. in the transgenic plants. And here what you see is the control, where the larvae is happily living and undergoing development, and they become pupa actually in the non-transgenic plants. So that's how the biopsies are performed, and the data which emerged from here, if you see, is in the control plant, if you see, where when we infected the plants at two different stages, the booting stage and the drilling stage, you know, larval mortality was nil, in the sense that the larvae were not killed, and it led to 50% of the damages, 50 to 55% stages. I mean, the damages, you know. And when you look at the transgenic lines, you know, mortality was seen 100%, 80 to 100% at the and the tiller damage was nil here, and 20% was the tiller damage in the transgenic. So from 50% damage to nil, you know, in the transgenic plants, shows that these lines are able to protect the plants 100%. There is no damage. You know. So, so what we have seen here, you know, that the technology which is being used here, a cryokinase gene, expressed into rice, and transgenic plants were able to protect themselves 100% as against the normal plants or untransformed plants, where the damages are 50% or so. So you imagine where a crop was damaged 50%, this technology could save that damage, and which is a lot in terms of agricultural performance in the field. So, how safe is this technology? As I mentioned earlier, that it is already used as a pesticide. You know, only thing is what we are trying to do is instead of using whole bacteria, we are using very specific genes genes to make the insecticidal protein within the plants. You know, and also it is eco-friendly or environmental friendly. It doesn't harm non-targeted insect pests, you know, which need to grow on Earth, you know. So that is one thing. Eco-friendly. How it is eco-friendly? Because of as I mentioned, it doesn't harm. It doesn't harm the insects which are harmless to the crop. It very specifically targets, say, in case of rice, the stem borer insects. You know, so it is safe. And then, as the concluding slide, this is what: protect non-stem borer and leaf folder by supporting the cultivation of paddy rice and reduce pesticide use. Okay, so that's the message I want to give towards the end, that it will be good if we use this technology, as I mentioned in the beginning, in my introductory lecture, that use of GMO is discouraged by most of the governments, and particularly in India, it is not approved. Other than the BT cotton, there is no other GMO which has been approved for commercial use. Okay, but nevertheless, these are the scientific efforts which need to be done, you know, and we need to keep on trying, that one day we might need these kind of products to be marketed. So with this, thank you very much for viewing this presentation.